Okay, so welcome to this video. In this video, what we're going to talk about is the adenylyl cyclase protein kinase C pathway. Okay, which is the pathway by which we activate adenylyl cyclase enzymes, which then produce cyclic AMP to lead to the activation of protein kinase A. So I'll call it the adenylyl cyclase / protein kinase A pathway. And often people use as an abbreviation for adenylyl cyclase AC, and for protein kinase A they'll use pKa. So you might see this referred to as the AC pathway, or the AC-pKa pathway, or maybe just as the pKa pathway. Okay, all of those names refer to the same thing. Some people will even call it the cyclic AMP-dependent pathway. Okay, right. So, the structure for this video then, we're going to start off by discussing G-protein coupled receptors, because it is G-protein coupled receptors which will activate the adenylyl cyclase protein kinase A pathway. Okay, we'll then discuss heterotrimeric G-proteins and the G-protein cycle. Then what we'll move on to is adenylyl cyclases, and then finally protein kinase A. Okay, and then we'll see what protein kinase A does. Okay, right. So we'll start off then with G-protein coupled receptors. Right, so we'll start off with the basic structure that all G-protein coupled receptors share, and we'll go over a little bit of nomenclature. Okay, so G-protein coupled receptors are a massive family of receptors. They are one of the biggest families of proteins known. Okay, in humans alone we have 800 different G-protein coupled receptors. Okay, and for short they're often abbreviated to GPCRs. And the characteristic feature that all G-protein coupled receptors share is that they all have 7 membrane-spanning alpha helices. Okay, so if these two lines represent the inner leaflet of the phospholipid bilayer and the outer leaflet of the phospholipid bilayer, so the inner layer of phospholipids and the outer layer of phospholipids, then I'm going to draw the polypeptide as it spans through the membrane. So they're one polypeptide, they're not, complexes of polypeptides this time, they are just a single polypeptide. Okay, right. So the polypeptide always starts with its amino terminus extracellularly. Then it spans the membrane, and the line, remember, is representing a polypeptide. It's representing a polymer of amino acids. It represents amino acid after amino acid after amino acid after amino acid, etc. Okay, so we have the first membrane-spanning alpha helix, then we have the second membrane-spanning alpha helix, the third, the fourth, the fifth, the sixth, and then the seventh. Okay, and then we have the carboxylic acid terminus intracellularly. So this is the characteristics feature that all G-protein coupled receptors share. They have 7 membrane-spanning alpha helices. They start with their amino terminus extracellularly, then they have this 7 membrane-spanning alpha helix domain, and then they have their carboxylic acid terminus intracellularly. Okay, so let's go over a little bit of nomenclature for the different portions of G-protein coupled receptors then. Okay, right. So we'll start with the 7 membrane-spanning alpha helices, which I'm now colouring in turquoise here. Okay, so even though I've drawn these just as lines, in actual fact they will be alpha helices. Okay, so the polypeptide will actually spiral round in an alpha helix like so as it spans the membrane. Okay, right. So these 7 membrane-spanning alpha helices are so important that they're given their own names. Okay, so the first one is called TM1, standing for transmembrane alpha helix 1. Then we have transmembrane 2, TM2, the second membrane-spanning alpha helix. The third membrane-spanning alpha helix is then called TM3. The fourth one is called TM4. The fifth one is called TM5. The sixth one is called TM6. And then the seventh one is called TM7. Okay, so this is TM7. So we have TM1 through TM7. I've only labelled the first and the seventh because it would get awfully crowded if I labelled up all 7 of them. Right, then we have names for these loops that are in between the membrane-spanning alpha helices. Now, 3 of these loops are intracellularly, so we have one here between the first and the second membrane-spanning alpha helice. We have another one here between the third and the fourth, and another one here between the fifth and the sixth. Okay, and these are known as the intracellular loops. Okay, so all 3 of these are intracellular loops. And for short, intracellular loops are often abbreviated to ICLs. Okay, so the I is for intra, the C is for cellular, and the L is for loop. Okay, so this first intracellular loop here between TM1 and TM2, this is known as ICL1. The second one here between TM3 and TM4, this is known as ICL2. The third one here between TM5 and TM6, this is known as ICL3. Okay, so that's the naming of those intracellular loops. We also have names for the extracellular loops here, which I'll highlight in blue. Okay, so once again, these are known now as the extracellular loops, or the ECLs. Okay, and again they're named ECL1 all the way through to ECL3. So the first extracellular loop between TM2 and TM3, this is called ECL1. The second extracellular loop between TM4 and TM5, this is known as extracellular loop, or ECL2. And the final one between TM6 and TM7, this is called ECL3. Okay, so that's the naming system for these intracellular and extracellular loops. We also then have the amino terminal domain here, which is this portion that's prior to TM1. Okay, so this portion is called the amino terminal domain. Okay, sometimes abbreviated to ATD, or even sometimes people call this the A-TED, like so. And what this stands for is the amino terminal, and then the E is for ectodomain. Okay, so let me write this out in full. So we'll have amino, and then the T is for terminal, and then the ED this time is for ectodomain. Okay, so ecto means on the outside, so this is a domain that's on the outside of the cell membrane, basically. Okay, and then this portion will be called the carboxylic acid terminal domain, or the C-terminal domain, CTD for short. Okay, right, so those are the different portions of G-protein coupled receptors. Okay, so as I say, there are a huge plethora of different G-protein coupled receptors just in humans alone, okay? Now, they will all have their own different ligands, and when those ligands bind to the G-protein coupled receptor, it will trigger a conformational change in the G-protein coupled receptor that will be conveyed down to these 3 intracellular loops. So the 3 intracellular loops will change in conformation, and that will now make available a binding site that is formed by these intracellular loops for the next protein along in this signaling cascade, which is going to be a heterotrimeric G-protein. So we now need to turn our attention to the heterotrimeric G-proteins. Okay, right. So, heterotrimeric, what does that mean? Well, basically, trimeric means that these are going to be made up of 3 subunits. The hetero means different. So that tells us that these 3 different subunits that are going to make up our G-protein are going to be 3 different subunits. Okay, so let's look at the 3 different subunits that make up our heterotrimeric G-proteins. And there should be a dash between G and protein. Okay, right. So the first of the 3 subunits which makes up a heterotrimeric G-protein is known as the alpha subunit. So we'll begin by discussing the alpha subunit. Okay, so I'll draw my alpha subunit here. Okay, now alpha subunits are special because they can be in 2 states. Okay, they can be in an on state. Okay, and in the on state they have guanosine triphosphate, GTP, bound to them. Okay, and they can also be in an off state in which they have GDP, guanosine diphosphate, bound to them. Okay, so depending on whether the alpha subunit has a GTP molecule bound to it or a GDP molecule bound to it, it will either be in an on state where it will be activating all sorts of signaling cascades, okay, and we're going to look at those, or it can be in the off state where it's not doing that. Okay, right, so I'll colour in my alpha subunit in red here, or at least I'll outline it in red. So let's start with our alpha subunit in the off state. Okay, so in the off state it's going to have a molecule of GDP, guanosine diphosphate, bound to it. Okay, right, the other thing to say about alpha subunits of heterotrimeric G-proteins is that they are not integral membrane proteins. They are not sitting in the membrane. However, they are attached to the cell membrane. They're attached to the underside of the cell membrane. Now, how does this work? Well, basically, attached to the polypeptide, you have a large lipid moiety. In some cases, you have two, okay? And I would just like to talk a little bit about this lipid moiety. But firstly, let me talk about the basic concept. The basic concept is this, that you're going to take this alpha subunit polypeptide and you're going to stick this large lipid moiety onto the side of it. And this large lipid moiety will be very hydrophobic. And basically it will want to implant into the inner leaflet of the phospholipid bilayer. So it's going to end up implanted into this inner layer of phospholipids of the phospholipid bilayer. And that's going to hold the alpha subunit dangling underneath the cell membrane. So the alpha subunits are not sitting within the cell membrane, but they are attached to the cell membrane via these lipid moieties. So let me give you the main example of lipid moiety that is going to be attached to alpha subunits of heterotrimeric G-proteins. Okay, so the main way that you attach a lipid moiety onto these alpha subunits of heterotrimeric G-proteins is for a process known as S-palmitoylation. Okay, and this involves sticking palmitic acid molecules onto sulfur atoms within the alpha subunit's polypeptide. Okay, so let me firstly tell you what palmitic acid is. Okay, so palmitic acid is the old name for a molecule that would now be called hexadecanoic acid. Okay, so palmitic acid is the old name for what's been renamed hexadecanoic acid. And although hexadecanoic acid is a slightly longer name than palmitic acid, it's a better name than palmitic acid because it tells you exactly what we are dealing with here. It's telling us that we are dealing with a 16, that's the hexadec. Okay, dec means 10, hexa means 6. So overall 16 carbon, fully saturated carboxylic acid. Okay, so let's draw that here. So, here is our carboxylic acid group. Okay, that's the first carbon here. We now need another 15 carbons. So 14 of these are going to be in the form of methylene groups. Now, I haven't got the space or the inclination to draw 14 methylene groups. So instead what I'm going to do is I'm going to draw one methylene group here, and then I'm going to put brackets around it like so. Okay, and then I'll subscript it down here, 14. Now, that's a useful trick to get around having to do a lot of work, okay? What you can do is just draw this one group that you want, and then draw these brackets around, subscript it with the number of them that you want, and this means repeat this structure 14 times, basically. Okay, so that now takes us up to the 15th carbon. We've got the first one here. We've then got another 14 in the form of these 14 methylene groups. And then the final one will be our 16th carbon. Okay, and the final one won't be in the form of a methylene group, which is why I can't draw it in this brackets. Okay, instead it will be in a methyl group, and it's good practice to, instead of trying to include this one in the brackets, to draw it separately. Some people will instead say, we have this 15 times, and then right on the end we just have a hydrogen. Okay, it's better, well, it's seen as better practice to draw the methyl group separately. Okay, right, so that's a palmitic acid molecule, or a hexadecanoic acid molecule. Okay, what we are going to do is we can attach these molecules onto the sides of proteins, basically, by attaching them to sulfur atoms within cysteine residues of the polypeptide. Now, what this will do is it will mean that we have this long, dangling 15-carbon tail of the carboxylic acid dangling off the side, basically, which is what this is. It's a very hydrophobic structure dangling off the side of the protein that's going to then implant into the inner leaflet of the phospholipid bilayer. Okay, so let me now just discuss how we're going to attach these palmitic acid molecules onto cysteine residues within proteins. Okay, right, so let's draw a cysteine residue of a protein. Okay, so we'll start with our core amino acid structure. So here's the amino group here, here's the alpha carbon with a hydrogen coming off it, here's the carboxylic acid group down here. Okay, so this is the core amino acid structure here. Okay, as it would appear within a protein. So I'm not drawing a pure amino acid here, instead I'm drawing a residue. Okay, so when people say they're drawing an amino acid residue, what they mean is that they're drawing the amino acid as though it is within a polypeptide. Okay, so the R group of a cysteine residue then is a methylene group like so, with a thiol group sticking off the side. Okay, so when you have a sulfur atom bound to a hydrogen atom like so, this is called a thiol group. Now, thiol groups have, to an extent, similar chemical properties to alcohol groups. Now, the reason for that is that sulfur is in the same group of the periodic table as oxygen. Okay, so it has the same number of outer shell electrons as oxygen. So, in this thiol group that we've got here, if that was an oxygen rather than a sulfur, that we'd call that an alcohol group. But it's a sulfur atom instead, but sulfur has similar chemical properties, so this thiol group, as it's called, is therefore going to have similar chemical properties to alcohol groups. Now, imagine for a second that that was an oxygen atom there rather than a sulfur atom. Then how could I bind a carboxylic acid group onto that alcohol group? Well, I'd perform an esterification reaction. Now, basically, thiol groups can react with carboxylic acid groups in exactly the same way as alcohol groups would to form a link that's very similar to an ester link. Okay, right. So, what's going to happen basically is, just as it would in an esterification reaction, what's going to happen is we're going to take the hydrogen off the, you can imagine this is an oxygen atom if you like, we're taking the hydrogen off the alcohol group, we're taking the alcohol group off the carboxylic acid group, we'll combine those two things together to make water, so this is going to be a condensation reaction, and then we're going to bind this carbon atom here of the carboxylic acid group to this oxygen atom here. And that would be an ester link if indeed this was an oxygen atom. We now have to remember that this is a sulfur atom, but this same reaction can occur basically just as it would if that was an alcohol group down there. Okay, and this link then that you've got where you have the palmitic acid molecule linked to the thiol group of the cysteine residue, this is called a thioester link. Okay, so when you link carboxylic acid groups to thiol groups, the link that you get is called a thioester link. Okay, so this is how you can attach palmitic acid molecules onto proteins. You can attach them to the cysteine residues within the polypeptide via these thioester links. Okay, and this is why it is known as S-palmitoylation. Okay, so firstly let me just tell you what a palmitoyl group is. So what is the difference between palmitoyl and palmitic acid? Okay, so basically you will notice that the portion of the palmitic acid that we have added on is not the entire molecule. Okay, so we don't quite add on the full palmitic acid molecule. Instead, we don't add this alcohol group here. Okay, so the portion we add on is this portion that I'm now circling in green. Okay, we add on all of the palmitic acid except the alcohol group that comes off the carboxylic acid group. This is known as the palmitoyl group. Okay, so it's not a molecule because this carbon has nothing. Okay, this carbon needs something else attached to it here, so it's a group rather than an entire molecule. So when you stick this group onto another molecule, such as this sulfur atom here, you have stuck a palmitoyl group onto that sulfur atom, and the process of sticking palmitoyl groups onto things is known as palmitoylation. Hence, this is S-palmitoylation, because we are sticking a palmitoyl group onto the sulfur atom of a cysteine residue within our polypeptide. Okay, so that's how you can add palmitic acid molecules onto alpha subunits of heterotrimeric G-proteins, and this will happen for all alpha subunits, okay? We're going to see another lipid modification that can occur for certain alpha subunits, okay? And those special alpha subunits are going to get these two lipid modifications occurring to them, but we'll have a look at that in the next video.